So it's, I think we're going to start this first conference of this series, and we are extremely happy to have Jim Rothman here with us for almost 1 month, as you know. And indeed, I mean, it's a bit stupid to introduce someone like Jim, but I will do it very briefly. Jim defended his PhD in Harvard, I think. And then he went to MIT with Harvey Lodish, where we could have met, because I was at MIT at the same time with Rajbandari, Uttam Rajbandari, in the chemistry department. And then you moved to Stanford and moved many places. I mean, Sloan Kettering, Columbia, and now Yale. And indeed, his work is very famous, has been recognized by a huge series of awards, including the Lasker Prize, including the Kavli Prize, including the Lansbury Prize. I think you got it at here, right? Did you receive it in France? And indeed, the prize with Randy Shackman and Thomas Sudoff in 2013 for the work on, well, I would say vesicular traffic, vesicular formation, transport, and fusion, and their importance in the functioning of the nervous system. So, Jim, it's a great honor for us, and I hope you will be happy here for 3 weeks, and that will give you the idea of coming back as soon and as often as you want. This house is yours.
Thank you. I have my own microphone.
Yeah.
Alain, thank you very, very much for a generous introduction. And it really is my honor to join you today. Can you hear me okay in the back? The microphone is on? Great. Okay. Well, also I would like to congratulate the Collège de France for its recent selection of Alain as its new leader. Also, I would like to express my condolences to Alain for exactly the same reason. I'm sure that the Collège de France will prosper as a result of this event, although the science of developmental biology may suffer for the next few years. So, but thank you for being my host over the next several weeks, which I'm very much looking forward to. Well, tonight, this is the first of 4 lectures. This one, as you can see, the lecture today will be focused on how proteins are sorted in the cell, and specifically how this gives rise to the 3-dimensional organization of the cytoplasm. Next week, I believe on Monday, I will talk about the logic of the Golgi apparatus, which is an organelle that many of us have been fascinated with for many decades. It performs an arduous task for the cell. It actually purifies proteins. In a way, it operates like a distillation tower that purifies petroleum into its various fractions. Its cisternae act like plates in a distillation tower, and I will offer to you evidence to that effect and some hypotheses concerning the still somewhat enigmatic function of this ubiquitous organelle. On the 23rd of September, the topic will be the mechanism of neurotransmitter release, and I'll describe recent studies, many, in fact, most still unpublished, partly because we haven't submitted them, but also partly because we've tried to get them published but haven't entirely succeeded yet, but we will. Studies of the mechanism of neural transmission, a topic with which I've also been concerned for many years. And the thrust of the issue there will be to try to explain, to understand how the fusion machinery, which I will describe tonight, the snare proteins, how the same machinery can operate on 2 drastically different timescales. A leisurely timescale for the processes that I'll describe to you today, occurring in a matter of seconds to tens of seconds, and an extraordinarily rapid timescale, 10,000 times faster than this, happening at every nerve ending every time you think a thought? How can the same molecular machine operate on two different timescales by a factor of 10,000? Still a mystery, but one that we hope is becoming accessible to current technologies. And then the last lecture on the 2nd of October will be meant really for the general public and anybody else that might be interested. What I'll try to address is the role of science in society. Why should society support science? What is it that science does for society? What is it that societies need to do to sustain innovative science and therefore themselves be innovative and economically energetic? And I'll take a historical approach, looking back on the development of a very important class of drug that controls the cholesterol in the body over a 50-year process. It has had an enormous impact on public health and also on economics of societies. It's led to the development of the modern pharmaceutical industry. And what I'd like to do is track historically how those developments were the product of innovative societies, starting initially in Europe, moving to the United States as a result of the dislocations of World War II, and how the lineage of the scientists who contributed formed a scientific culture that could be preserved because certain societies chose wisely in how they fostered innovation. And finally, what I'll try to do is open a discussion on whether we're doing a good job today, we being Europe, we being the United States, and we being increasingly China. So why don't I get started with tonight's lecture. And what this is the story of really almost a half a century of discovery recognized by three successive awards of the Nobel Prize in medicine or physiology over this period of time. And it's the story of the itinerary of newly made proteins in the cell. And at a very sort of high level, for those of you who may not be biologists, the cell is complex anatomically. It has many different locations called organelles. And each of these locations has its own unique composition, a unique set of proteins specified by their own genes. And each of these proteins, each of these sets of proteins, provides for a unique set of functions. So to be very abstract about it, and not at all concrete, let's imagine that the cell has perhaps 20 different locations, but let's just talk about perhaps 3 of them, A, B, and C. A has its own set of perhaps 1,000 different components. B has its own set, largely non-overlapping, of different protein components. C, its own set. So we have A proteins, B proteins, and C proteins. Now, those proteins, like all proteins in the cell, are made by a particle called the ribosome, a multi-enzyme complex. And the key point is that the cytoplasm has only one kind of machine, one kind of ribosome, and it must turn out all of the different proteins in the cell from whatever gene it happens to be expressing. I seem to have unplugged my plug, which is probably not a good thing in a lecture. So the consequence of this simple fact is that you have one protein manufacturing machine, the ribosome, and it synthesizes A, it synthesizes B, and it synthesizes C. So by definition, A, B, and C are all produced as a mixture in the same place, the compartment that contains the ribosomes. Now somehow, A must be physically separated from B and from C. Proteins A must be recognized by something and dispatched or sorted to destination A, B to B, and C to C, and D, and E, and F. It's an enormously complex process, and it occurs in real time in virtually all of our cells in our body. So that's the problem, and we call that the sorting problem. And over the last several decades, as a result of the research I'll now describe to you, we've come to an understanding at almost, I would say, almost an atomic level of detail of many steps involved in this process, which was entirely unknown as a process before about 1950. So that's where this story begins with George Palade, who did the bulk of his work at the Rockefeller University in New York, and later in his career moved to Yale, where he founded the Department of Cell Biology at Yale Medical School, and was its first chair. And as a matter of fact, I am now the present occupant of that chair of cell biology. So there's a certain lineage in that respect. Now, Palade was among the very first to recognize that the electron microscope could be utilized for biological purposes. And he and his colleagues at the Rockefeller learned how to use the electron microscope, how to make thin sections of cells, how to stain the components of them, to reveal the membranes, and in the process, they discovered the modern ultrastructural version of the principal organelles in the cell, the nucleus, the envelope of the nucleus, and especially the complex organization in the cytoplasm. And in the process, they discovered the ribosomes. Actually, I believe it was Palade who named the ribosome, and it was he who suggested that it makes protein. Later, scientists would prove that and work out how. He discovered the membranes inside the cell. He discovered that these membranes each form compartments. Now, I've already mentioned compartments A, B, and C, etc., as concepts. In reality, each compartment is a membrane-enclosed space, enclosed by a membrane that is very thin. The thickness of this membrane is perhaps 40 angstrom units, okay, 4 nanometers. That's how thin it is. And it completely surrounds a space. A different but related membrane will surround another compartment, and so on. So we might call this compartment A, that compartment B, and that compartment C. Unfortunately, it doesn't seem that this light works all that well, but I think we can make do. Can you see it at all? No. Nope, not really. How about if I keep moving it? Well, a little bit. Okay, I'll use this. So this compartment is George Palade and his colleagues, especially Keith Porter, called the endoplasmic reticulum. And you'll note that the endoplasmic reticulum has ribosomes bound to it. Those ribosomes are making the proteins for A, B, C, and D, and they are being introduced as a mixture into the lumen of the endoplasmic reticulum. So that is where the sorting process begins. Then you'll notice these cisternae that are arranged in a stack. This is the Golgi apparatus, whose ultrastructure Palade also discovered. And that will be the subject of my lecture on Monday. You will also notice there are other organelles in the cell, of which one is membrane enclosed, called the condensing vacuole. And you'll notice that there are smaller objects that are called Golgi vesicles. Now, what Palade did was provide an observant eye to categorize these structures in their appearance in the electron microscope, but he also invented an entirely new technique that allowed him and his colleagues to trace the pathway of these newly made proteins for the first time. And it is called autoradiography. And he incorporated radioactive amino acids by incubating, in this case, slices of the pancreas, because the pancreas, of course, is engaged in secretion. The purpose of the pancreas, in part, well, mostly, is to secrete proteins. It secretes insulin into the blood, and it secretes digestive enzymes into the GI tract, the gastrointestinal tract. And in particular, the cell that Palade studied secretes a series of digestive enzymes. He labeled these newly made enzymes with radioactive amino acids, which were incorporated into the newest proteins. And then he and his colleagues prepared very thin slices of these sections of the pancreas. And then he put a film over the top of them that could detect the radiation. Okay? And then in the electron microscope, they could see where these proteins actually were. And what they discovered is, for the first minute or two of their life, the newly synthesized the digestive enzymes were found within this endoplasmic reticulum, which, because of its studded nature of these ribosomes, was called rough endoplasmic reticulum. But if he waited 10 minutes, the radioactivity now moved to the Golgi apparatus. And if he waited perhaps another 10 minutes, they moved to these condensing vacuoles. And if he waited even longer, these vacuoles would merge with the cell surface in a process. Thank you very much. Thank you also, Alain. I think. Well, let's find out. Yes, if you know how to use it. Perfect. Thank you. And these condensing vacuoles, moments later, will merge with the surface of the cell to release the digestive enzyme into the gastrointestinal tract. So in short, what Palade had discovered was the secretory pathway. He discovered the principle that this organelle, the endoplasmic reticulum, is where proteins are made that will later be secreted from the cell. And in the fullness of time, we learned that these proteins included, of course, the digestive enzymes, hormones like insulin, growth factors, and eventually we learned in the brain they included neurotransmitters. So this was a major discovery in the history of biology. He also discovered that these proteins must journey from where they are made to where they will ultimately end up, and that they do so in a series of stages. First to the Golgi apparatus, and later to a storage vesicle, which holds back the secretory cargo until the time is ready for its release into the digestive tract. Again, an important general principle, a secretory storage vesicle. Now, what Palade's work also did was to open a number of questions. And those questions have gradually been resolved and continue to be resolved in the following years. By discovering this pathway, he raised the question, how general is this pathway? Because after all, there are many other compartments in the cell besides the outside of the cell where the secretions are directed. There is the Golgi apparatus, the plasma membrane has its own proteins, endosomes, lysosomes, and again, in the fullness of time, we learned that an entire range of proteins, A, B, C, D, E, F, likewise are synthesized by these ribosomes on the endoplasmic reticulum, are carried through the Golgi apparatus, and only there are they branched off and delivered to their various locations. So we learned, as Palade described in his Nobel lecture, that there are variations on this pathway that are essentially needed for the growth and development and differentiation of the cell, not to mention its continued function. He also suggested a mechanism, and this was the other question raised, how are proteins carried among these organelles? It's an interesting problem because each organelle is a self-contained membrane-bounded unit. So how does a protein located like this X within the endoplasmic reticulum, how is it transported, for example, into the Golgi apparatus or into a condensing vacuole? What is the basic idea? Well, the basic idea that Palade suggested is that little vesicles could bud off, they could simply bleb off from one of these membranes, somehow travel over, and then join with a destination membrane. We call this the donor membrane, we call this the acceptor membrane or the acceptor compartment, and this transport vesicle would somehow package the cargo, X, carry the cargo through the cytoplasm, fuse with the destination or acceptor compartment, thereby incorporating the cargo and moving it vectorially from one location to the next. This was the general idea that Palade introduced, okay? But just an idea. Now, this is an example of what George saw. This is actually one of his original micrographs from the early 1960s. This scale is 200 nanometers. So each of these objects, this is actually one of the vesicles that he saw. To give you a sense of scale, that is 70 nanometers in diameter, very small. A nanometer is 10 angstroms. An angstrom is the size of a hydrogen atom, more or less. So this is only 700 or so hydrogen atoms in size, so it's very, very small. This is the rough endoplasmic reticulum. There are the elements of it. That's a ribosome making protein. This is a vesicle budding. A few seconds later, it might very well become this vesicle. These vesicles travel over to the Golgi apparatus where they join with it and they fuse. This is a vesicle that a moment ago, this could be a vesicle that a moment later is fusing. Of course, you cannot visualize the time sequence in electron microscopy, but in the fullness of time and with all the biochemistry that has emerged, we know, in fact, this is what had happened. This is a condensing vacuole. You'll notice the secretion is very dense within it. And here happens to be another organelle. This is called a multivesicular body because of the many vesicles contained within it. This organelle is involved in digesting and turning over other organelles for fusion with lysosomes for degradation. It's just part of the cellular economy. That would be an example of compartments F, G, and H, in this case discovered, many of them, by Christian de Duve, who shared that Nobel Prize with Palade in the mid-1970s. Okay, so this is the pathway of intracellular protein transport. And when what Palade left us with was an understanding of a problem, a very profound and broad understanding, but what we had at the end of the day were static images in the electron microscope. How could this work? How is it possible that this could work at a molecular level? The first important step was taken by Günter Blobel, also at the Rockefeller University, and a student of Palade, who was given the opportunity to start his own laboratory there. And when he did, he decided that the right way to start would be to study and understand these ribosomes, because at that time, in the late 1960s and early 1970s, it was possible to purify this rough endoplasmic reticulum membrane as what were termed rough microsomes. They could be isolated from the other cellular organelles, from homogenates, because of their density, which was higher than most membranes because the ribosomes attached to them are physically dense, and they made that membrane dense enough to isolate effectively in pure form by centrifugation. Also, much progress had been made on the genetic code. Messenger RNA was discovered, and ribosomes had been isolated and shown to engage in cell-free protein synthesis accurately in cell-free systems. So it was a great starting point. And what Blobel sought to understand was, how could a ribosome know what protein it was synthesizing? Because I actually lied to you a moment ago. There were really two types of ribosomes in the cytoplasm. They're chemically the same, but they're functionally different. One is free in the cytoplasm, like these, and the other is bound to the endoplasmic reticulum membrane. The ones that are free in the cytoplasm are actually manufacturing, mostly, proteins that will remain in the cytosol, enzymes of intermediary metabolism and the like. The ones that are localized to the membrane, as Blobel and Sabatini discovered, are engaged in the synthesis of proteins that will be exported in the Palade pathway, secretory proteins and proteins of the plasma membrane. So there must be a mechanism that allows a ribosome to somehow know what messenger RNA it is translating, and then to choose accordingly where to go. The endoplasmic reticulum, if it's making A, B, C, D type proteins, to be exported, initially to be imported into the endoplasmic reticulum lumen, and then to be exported to other locations. Or, let's say, type 0 proteins, none of the above, that have no special property and therefore remain in the cytosol. And what Blobel discovered were the signals that these proteins have that direct them in the cell. Specifically, he discovered that each of the proteins destined to the endoplasmic reticulum, remarkably enough, A, B, C, D, and E type proteins, all have at their very beginning a special signal peptide that, if not identical, is very, very similar among all of these proteins. And if you think about that in the era before genome sequencing, just the idea that each of these proteins has a common sequence is an amazing fact. That's why this discovery was amazing. There are perhaps 25,000 human genes, okay? Perhaps 25% of them, roughly 5,000 or 6,000 genes, are proteins that go through the endoplasmic reticulum and on to these other destinations. It's an enormously complex process. Of course, not every cell makes all of them, but in the body, we make all of them. And so how can you have 5,000 genes, each of which has essentially the same sequence inserted at exactly the right place at the beginning of the gene? It's an amazing fact, okay? And it really speaks to the power and versatility of molecular evolution. In any case, Blobel discovered this by translating the messenger RNAs encoding this type of protein, A, B, C, D, from those, and comparing those proteins as a group to those that stay in the cytosol. And he discovered that these secretory proteins all had these N-terminal extensions, which were effectively removed by an enzyme located on the inside of the endoplasmic reticulum, explaining why the final product of the proteins never gave a single sign of this signal sequence. He generalized this concept to the idea that every protein, A proteins, B proteins, C proteins, D proteins, etc., each one has to have such a signal, and the purpose of that signal is to get it into the endoplasmic reticulum. And he further generalized the idea that to the idea that at every stage of transport, each protein must contain its own special signal to route it through the organelles so that each type of vesicle can recognize the right cargo protein. And that, in the fullness of time, turned out to be correct in the hands of others. So the signal hypothesis was really a landmark in cell biology, and it solved for us the problem of how A, B, and C are delivered as a mixture into the lumen of the endoplasmic reticulum. Okay? In subsequent work, Blobel discovered the machinery that translocates the proteins, called the translocon. He first proposed its existence, and he and others were the first to purify it and demonstrate its activity in highly simplified test tube systems. So this is well understood biochemically, and in more recent years, Tom Rappaport and his colleagues have provided us X-ray crystal structures that have given us a precise mechanism of how the signal peptide gates the channel and opens it to allow the subsequent polypeptide to go through. So a remarkable series of accomplishments, which, of course, as in every great piece of science, only opens the door to the next level of questions. And the next level of question was in fact defined by Palade himself, which is how can these proteins, now we understand how they arrived as a mixture in the endoplasmic reticulum, how can they be physically transported? And is it true, as Palade suggested, that these vesicles, these small 70 nanometer vesicles, are really responsible for that transport? Because the time resolution of the techniques available to Palade and the spatial resolution did not allow that distinction to be made. They allowed it to be suggested as a reasonable hypothesis. Okay, enter Randy Schekman, with whom I shared the Nobel Prize in 2013, and whose name has already been mentioned. Now, what Schekman did primarily was to identify in yeast a large series of mutations, initially 24 of them, and subsequently his students and others following his lead have identified a great many more temperature-sensitive and other mutations that specifically block various steps in Palade's secretory pathway. Some mutations block transport from the endoplasmic reticulum to the Golgi. Others block transport from the Golgi to the cell surface. Either one blocks the secretion of proteins from the yeast cell. Likewise, other genes block transport from the Golgi to multivesicular bodies and the lysosomes that I've mentioned. So what Schekman discovered, therefore, was, of course, that the secretory pathway exists in yeast. It's really very general. He discovered that it's required for the growth of cells, an important conclusion. Much more importantly, though, it's governed by specific genes, and therefore there are molecular mechanisms and machinery that can, in principle, be discovered that must somehow be responsible. Now, an important finding that he made in around 1990 was that Some of these genes actually block endoplasmic reticulum to Golgi selectively. Actually, that was a discovery from 1980. But he could classify these genes in that some accumulated vesicles containing cargo en route to the ER to the Golgi, whereas other mutations would accumulate the cargo in the endoplasmic reticulum. And the first type of gene must therefore govern the fusion of the vesicle, and the second must govern the production of the vesicle. And when vesicles that fail to fuse accumulate with cargo, he could reverse the temperature and reverse the lesion, and these vesicles would now fuse with the Golgi, delivering their cargo. This actually provided the first proof of Pilates' hypothesis that vesicles are the intermediaries in the living cell that carry cargo between membranes. But it did not answer the question of how this vesicle forms, how this vesicle fuses, how it packages its cargo, or how it knows where to fuse. Because let me give you a sense of the immensity of the problem the cell faces. This is just, even in a yeast cell, one of perhaps 10 different types of vesicles. In our cells, we have perhaps 20 different types of vesicles carrying A, B, C, D, etc. through most of the alphabet. There are that many vesicles at any one time. Each of those vesicles lasts for a very short time, maybe 10 or 20 seconds, maybe 1 minute at the most. It is made, it travels, and it fuses. And when it fuses, it loses its identity. When that vesicle fuses, it ceases to exist. It's made, it exists, it no longer exists. It lives, perhaps, let's just say, arguably for 1 second, and there are perhaps at any one time 100,000 or 200,000 of these in the cell at a time. Each one lasts a few seconds, and there are a dozen different types. Can you imagine how confusing that is for the cell? And yet, each vesicle, every time, needs to go to the right place. Sometimes the targets are very near each other. This vesicle needs to go either here or over there. It cannot make mistakes very frequently. And yet, it has to make that decision in a second or two with absolute accuracy. 100,000 times a second. It's amazing, right? It's amazing. So how does that work? So that is where my story begins. And my story actually, in a way, like all of us, is dependent on when you grow up, okay? So I was born in 1950 in the United States in the midst of an enormously powerful era of development of physical technologies. Building on the great discoveries in the earlier half of the century in physics, principally in Europe, the technologies became available that were developed largely in the United States. These include atomic energy with its Einstein, that's Oppenheimer, with its two phases for uses that are very difficult and troubling, and uses which are still politically debated as to, for power and energy. It includes the Cold War and the response to the Cold War that led to space exploration and the enormously inspiring era that I grew up in which, with utter amazement, you know, it seemed like every week somebody was in orbit, and then next thing you knew, someone was on the moon. It was an astonishing era in which the physicists were the heroes. And so if you were a young man or a woman, and you were more often a man in those days, and you were growing up and you had an interest in anything scientific or quantitative, you almost always were going to become a physicist, because that's who the heroes were. They were actually, you know, there are polls where they, at least in the United States, who are the most admired. You may have them in France as well, I don't know. Lawyers, at least in our country, are always very low on the list. Politicians, perhaps a little lower. Doctors are always sort of high up. Pop stars and sports stars, pretty high up. But in my day, in 1960, the physicists were number 1, 2, or 3. Okay, Oppenheimer and Einstein were the best known. So this is what you got interested and excited about. Even at the age of 12, I was making rockets. That's me. That was one of my rockets. It went up, not far enough. But you know, that's what I thought, so I studied physics. I went to Yale University actually as an undergraduate, and I was a very avid theoretical physics student. Somehow in my last year at Yale, my father, who was a physician, convinced me that I really ought to learn some biology because after all, biologists, there might be some, you know, frankly, he thought I wouldn't get a job. And it would be good if I were a doctor, as he was, in case that happened. And so I wanted to please my dad, and I took a course in biology. Despite all of my physics colleagues who said I shouldn't, they said, you know, actually, you know, biologists are really stupid. Of course, that's not true, but in that era, maybe physicists still say these things, I don't know. Perhaps less these days. And we all know that biologists, at least we all thought we knew biologists are stupid. I thought maybe I would try some chemistry because chemists were considered to be stupid by physicists, but at least they were sort of in the right direction. You know, that made them socially acceptable, but perhaps not brilliant. Anyway, I took a biology course. That was the end for me. It was just so amazing. I ended up going to medical school as a medical student, and it was as a medical student that I learned of George Pilates' work and saw that electron micrograph that I showed you as a first-year medical student at the age of 20. And I thought it was so amazing. How could anything like that happen? Well, it became my life problem. I gave up physics. I got a PhD degree. I learned how to be a biochemist with Eugene Kennedy and later with Harvey Lodish. I learned how to study proteins. But I never forgot how to think like a physicist. And so the way that I approached this problem was very much, although it wasn't physics, I think very much the way a physicist thinks about a problem. So if you say, well, how does a physicist think about a problem? I think you can say there's the two key features. One, a physicist seeks universal laws, tries to explain as much as possible based on a common basis. Isaac Newton, the founder of modern physics, really. When we think of him, we think of the falling apple. But perhaps what we should think is this unbelievable concept that the behavior, measurable behavior on Earth can govern behavior in the heavens. What an amazing thought, okay? You know, just an amazing that anybody could have such a thought, never mind in the era in which he had it. Okay, well, that's the way physicists think. And always formulating the simplest hypothesis to explain the facts. So here you have a process, the Pilates pathway, of enormous complexity. And you have, in a way, what was the prevailing hypothesis when I took up the problem about 1978, when I started at Stanford University as an assistant professor in biochemistry. This is the way I looked at it, and this is the way everybody else, I think, looked at it. Anatomy dictates specificity to explain the pathway. What do I mean by that? Well, if you look at this picture again, you'll notice that the Golgi apparatus is sitting very close to the endoplasmic reticulum. You see that vesicle there that looks like it's budding? It is budding from the endoplasmic reticulum. It could easily travel over and it could hardly miss the Golgi apparatus. You'll notice that the Golgi apparatus has two surfaces to it. One end where the vesicles are delivered, and another, the opposite face of the stack, where the finalized cargo, like the secretion here, emerges, okay? That's called the entry phase or the mature phase. Today we call it the cis and the trans phase. Everybody thought that the anatomy was critical, that actually the endoplasmic reticulum was simply feeding the vesicles to the Golgi apparatus because it's nearby. That's how it could choose. It would never go to the condensing vacuole, which was on the other side. After all, it would have to get past the Golgi. Likewise, a vesicle coming from here could hardly miss the condensing vacuole. It would be like saying, I have an apple in my hand, and I want to feed it to my mouth. So I go like so. Okay? Anatomy dictates specificity. But there's another way of looking at it, which is the way a physicist or a chemist would look at it. Intrinsic specificity. That's a very hard thing to understand how that would work. But what if each vesicle had built into it a chemical address marker, a signal. And what if that signal had a receptor that was located only at the target compartment? What if each type of vesicle had its own signal that no other one does? So that each vesicle would, through a chemical interaction, simply bind to the correct target, and then a general mechanism for fusion without any loss of specificity could result in the fusion. Likewise, when the vesicle forms, what if it formed with a scaffold that contained its own receptors that received the cargo? Now, each vesicle would select its own cargo. A would go to coat A, B to coat B, C to coat B, each to form its own type of vesicle, A, B, C, and each vesicle would have its own code to go and stick to the right compartment. That is specificity dictating anatomy. It would be the equivalent to juggling the apples. Okay, now imagine I have apples and oranges in my hand, and I've only got and I want to put apples in my mouth, but not the oranges. Well, if it happens to be specificity by anatomy, I have one hand with an apple, it goes like this, another hand with an orange, and if this organelle is in the wrong place, my mouth gets both. Not good. On the other hand, if I'm juggling, and my mouth is meant to get A but not B, apples but not oranges, then the apples and the oranges get all mixed up in the cell, but if my mouth is a receptor, has the right shape for an apple but not an orange, the apple gets in. That's how I suggested that it worked. Anatomy, meaning specificity, dictates anatomy. The forms that we see in the electron microscope are emergent properties that emerge from a still unknown molecular underpinning. Okay? Now, why? This is actually a drawing from 1982, long before the machinery was discovered, that just sort of illustrates this different way of thinking. Now, why did everybody like this, and why didn't they like this? Well, they liked this because that's the way the cell looked. And the people in the field were actually anatomists in their tradition. Okay? People who were biochemists or chemists would naturally look at it this way, which is, of course, the way I did. And it is indeed the simplest hypothesis, which reminds me to introduce, even at this late hour, Edward Buchner, who is the founder of modern biochemistry. And why do I bring him up? I bring him up because this way of thinking, the idea that even the most complex biological processes in a cell are chemical in nature and therefore can be reproduced outside of cells in the absence of life as a purely chemical process, that basic insight, okay, is nothing new, at least for my generation, but applying it to this problem perhaps was new. What Buchner taught us was that fermentation can occur outside of a living cell. Fermentation, of course, was a subject of great delight and discussion in this city in the middle to late 1800s. And it was Pasteur who showed that life, and therefore fermentation, could only come from life. And it took a long time for chemists to appreciate, given the importance of that dogma, that actually processes, and it is true that life only comes from life, at least so far. We haven't made life from the test tube. But it's also true that processes that are responsible for life, like respiration, nothing is more vital, can occur outside of living cells. And this is actually what Buchner discovered in his landmark studies to show that broken up yeast cells made into a soluble extract could convert sugar to alcohol. Okay? Now, it's also one of the great stories of modern science, ancient science almost, 100 years old, but it's a great story because he was interested in more economical ways to make alcohol for fermentation, okay, and as a pharmaceutical. He did not expect to discover the underlying principle of biochemistry, and this is a quote from his Nobel lecture, which I absolutely love. We are seeing cells more and more clearly as chemical factories, where the various products are manufactured in separate workshops, the enzymes acting as the overseers. Do you know enzymes in those days were only known in terms of their ability to modify the color of dyes, and yet he related them to this process. It's really extraordinary. What an extraordinary vision. So the remarkable prediction of the simple hypothesis is that vesicle traffic, which itself generates anatomy in the cell, nonetheless can take place in a cell-free extract. So that's what I set out to test in 1978. Could we reproduce the accurate production and targeting of a transport vesicle in a cell-free extract when we had destroyed all of the existing cellular anatomy? And if we could do that, it meant that there had to be chemical specificity. Okay? And if there is chemical specificity, it meant there must be enzymes. And if it meant there must be enzymes, it would mean that we could purify those enzymes and characterize them rationally as to how they work, and then begin ultimately a journey to understanding the molecular and the chemical, and ultimately ultimately at an atomic level, the molecular principles involved in protein sorting. We succeeded in doing this in 1980, although the work was not generally accepted by my colleagues probably until the late 1980s. And what we did was we mixed two extracts from two different types of cells. And the reason for two types of cells is there can be no existing anatomical relationship of the inside of one cell to the inside of another cell. So it was a sort of conceptual point. And what we did was we have one cell whose Golgi apparatus was loaded with a cargo protein, and a second cell that lacks that protein. Now, the clever thing in the experiment is that this cell, the donor cell, is missing an enzyme that normally adds a sugar moiety, a sugar molecule, to this glycoprotein, also in the Golgi. This other cell, the acceptor cell, it has the sugar transferring enzyme, but it doesn't have the cargo protein. So when we mixed an extract containing these Golgi compartments with a separate extract from a different cell containing these Golgi compartments, the only way that this cargo protein could receive a sugar and end up within this membrane is if it was able to travel from one membrane to another in a way that allowed its distribution while retaining the intact nature and separate nature of these two organelles. In other words, if we could observe sugar, like radioactive sugar, incorporated into this cargo protein, it would require the formation of the vesicle that Palade had seen, the traveling of this vesicle, and the delivery of this vesicle. And the delivery would have to be very specific, because if it were unspecific, since these membranes are only 1% of the membranes in the extract, very little of the cargo could be delivered to that location. Well, of course, that's exactly what we observed. And this represents the efficient incorporation of transfer of about 50% of the cargo protein, about the theoretical limit, from the donor Golgi to the acceptor Golgi. And this transfer requires energy, ATP. That shouldn't be a surprise because it's vectorial. And it requires enzymes, the soluble high-speed supernatant. So this all looked very promising. At this point, I began a collaboration with the consummate electron microscopist, Lelio Orci, at the University of Geneva. And we also discovered inhibitors that could block this transport. And the inhibitors are essentially poisons. They're the biochemist's version of a genetic mutation. And what they do is, if you have a pathway like membrane to vesicle, to docked vesicle, to fused vesicle, if you block a specific step along the way, and now you look in the electron microscope, for example, I'll show you this in a minute. If you block fusion, but you allow the vesicles to be produced, what will happen? Vesicles will accumulate. You look in the electron microscope, even electron microscope images of these cell-free incubations, and you should see large numbers of vesicles accumulating. And that would establish the role of these vesicles as intermediates. Okay? Early on, we, and in this case I mean me, because I did these experiments, they were the last ones I ever did, I have to say, with my own hands, added non-hydrolyzable GTP to these cell-free extracts. And what we found is that transport was inhibited. So delivery of the cargo was blocked. But when we looked in the electron microscope, tons of vesicles had accumulated. And we were able to purify them. And this is a test tube full of pure transport vesicles. They're produced in the test tube. They contain the cargo protein I just mentioned. And if we mix them with another extract or another Golgi, they go on to deliver their cargo. So they are functionally competent transport vesicles. They contain within them the cargo protein, and you'll notice this conspicuous outer coating. That's called the coat that we postulated years before might be important in selecting the cargo and in helping form the vesicle. Well, now, having in hand these pure vesicles, we could run SGS gels, we could identify the components of the coat, and what we observed was that the bulk of the coat was made up of a complex of 7 proteins that comes from the cytosol. They stay together, and it's called the coatomer. This coated vesicle is now called the COP1 coated vesicles, and it is responsible for transport within the Golgi. The basic principle became clear because we could incubate Golgi membranes with and without coatomer, and without coatomer they wouldn't form these vesicles. And we found there was another protein called ARF that is needed together with the coatomer to form the vesicles. ARF by itself will not form vesicles. Coatomer won't form vesicles. You put the two together, you can form the vesicles, but only if you add GTP. ARF binds GTP. That causes ARF to localize to the membrane. ARF then binds the coatomer. It causes a conformational change, as Felix Villon discovered, of the coatomer, causing the coatomer to polymerize into a shape of a sphere, which then deforms the attached membrane, and the vesicle is thus shaped and is removed from the membrane. Receptors within the coatomer bind the cargo and are responsible for the selection of the cargo, as shown in later work by others. Importantly, this coat, while it is needed to form the vesicle, is now a roadblock for fusion. The vesicle inside needs to fuse, in this case, with the Golgi. How can it fuse if it is encased in a skeleton of protein, an exoskeleton? You needed the protein exoskeleton to shape the vesicle, but the skeleton now must be removed. How does that happen? That happens, we discovered, when GTP is hydrolyzed. When GTP is hydrolyzed to GDP, the ARF once again loses its affinity for the membrane, goes back to the cytosol, releasing the coat. That is why when we provided non-hydrolyzable GTP instead of GTP over here, we ended up with non-hydrolyzable GTP over here, and when it came time for the coat to be removed, it could not be removed, and therefore these COP1 coated vesicles, the intermediates, accumulated, which enabled us, in fact, to discover them. The coat had gone unnoticed by Palade and all of the workers for all those years until we discovered it in these test tube incubations, because it's such a transient intermediate. But now, by making it permanent, we could discover it, and we discovered the role of these vesicles in cargo transport. I'd like to bring you back to the second inhibitor we found, N-ethylmaleimide. This is also a poison. It's less specific, but interesting, in that when we poison transport with N-ethylmaleimide, and now we look, we also see the accumulation of large numbers of vesicles, but you'll notice these vesicles are all lined up at the target membrane, Golgi cisternae. They are fully formed. Do you see any coats on them? There are no coats. They've lost their coats. And they are ready to fuse, but they don't fuse. So what does that tell you? It tells you that there must be a protein inhibited by this chemical, which alkylates cysteines, sulfhydryl groups, that is responsible or otherwise contributes to the fusion of these vesicles. We call that protein N-ethylmaleimide sensitive fusion factor, NSF. And we were able to purify this protein from the cytosol of untreated cells by adding back fractions of protein to poisoned extracts and looking for the functional recovery of trafficking, therefore membrane fusion. And we purified that protein to homogeneity. We discovered it is an ATP hydrolyzing enzyme, thus accounting for the ATP requirement, at least in part, in membrane fusion. We also discovered that in yeast it is encoded by one of Randy Sheckman's genes, SEC18, which accumulates vesicles that are forming from the endoplasmic reticulum to fuse with the plasma membrane. That is a discovery he made somewhat later, but a very important discovery. And this connection between NSF, purified on the basis of this very artificial assay, and the gene that affects transport in living cells made it very clear that this pathway is universal, even in its details, and made it very clear that there must be a general role for NSF in membrane fusion. So how does NSF participate in membrane fusion? This was made clear with the discovery of the snare complex by Thomas Zollner in my laboratory in 1993. And what we did was we asked, how would NSF interact with the membrane? We knew that NSF hydrolyzes ATP. It's got to somehow interact with the vesicle or target membrane if it's going to be involved in fusion, but we didn't know how. So we imagined that it must bind. I left out an important step. We purified another protein called soluble NSF attachment protein, which is needed to attach NSF to the membrane. And then the question became, what is the receptor for the SNAP protein in the membrane, like the Golgi membrane? What is the nature of that SNAP receptor that then allows NSF and SNAP to operate? The SNAP receptor is abbreviated S-N-A-R-E, snare. That's where the word snare comes from. So we wanted to identify the nature of these snare proteins, which by implication would be important in membrane fusion. I should mention importantly that NSF and SNAP are found throughout the cell in the cytoplasm, and they are needed for every cytoplasmic membrane fusion process. So they power, with it somehow or other, with ATP, membrane fusion, but they do not provide specificity. They cannot because they work at multiple locations with A, B, C, and D type transport vesicles. What Zollner did was mix NSF pure protein with SNAP pure protein and an extract of the total membranes of an organ. As a result of that, he could isolate by immunoprecipitation against NSF the proteins that were indirectly bound to NSF by binding to SNAP protein, and which therefore are likely to be the SNAP receptor or snare proteins. And he found a very simple polypeptide composition. There were 1, 2, 3 species of proteins present in a 1 to 1 to 1 molar ratio. We called these 3 proteins the snare proteins, SNAP receptors, and we called the complex of these proteins the snare complex. And we suggested that the snare complex might be involved in membrane fusion. So this was the discovery of the snare complex. Now, as it happened, we didn't use any organ at random. We used brain. Okay? Why did we use brain? It doesn't matter. We were just lucky. And as a result of being lucky rather than smart, we also discovered, en passant, the mechanism of neurotransmitter release. You remember I said neurotransmitters are secretory molecules. Well, the brain is highly specialized to release neurotransmitters. And the most abundant membrane fusion proteins in the brain, are therefore snare proteins, are involved in releasing neurotransmitters. So the brain, like every tissue, has all of the many. There are probably 50 snare proteins encoded in the human genome. They're all there, but quantitatively the most abundant are those involved in neurotransmitter release. And so hidden under this, no doubt, and when we look for them, are many snare proteins, but particularly syntaxin, SNAP25, and VAMP. These proteins were known anatomically, but we didn't know from the work of Scheller and Sudhoff and Jan and others. We didn't know what they did, but it was very important that the work they did had been done. It was very important work. And the synaptic vesicle contains this protein, vesicle-associated membrane protein, which should give you some idea from the name. We didn't know what it did. Syntaxin, also found in the plasma membrane. Synaptosome-associated protein 25 kilodalton, not actually even known to be in the plasma membrane. And finding this complex suggested that there was a binding between a membrane protein of the synaptic vesicle, this vesicle-associated snare protein, or V-snare as we called it, and these proteins in the plasma membrane, the target membrane for fusion. We call these T-snares, V-snare, T-snare, and we suggested that all cells and all membrane vesicles, types A, B, C, and D, each have their own type of snare, each vesicle its own V-snare, each target membrane its own T-snare. And so to bring you back to the original idea, Searce in 1980, this is now 1993, we suggested the snare hypothesis that each vesicle is encoded with its own signal or address marker. That signal would be the V-snare itself. In the case of the synaptic vesicle, that V-snare is the molecule VAMP, or a similar molecule. In the case of the endoplasmic reticulum to Golgi vesicle, or an intra-Golgi COP1 vesicle, it would be a different molecule, but structurally related. That would be the V snare. The plasma membrane would have its own T snare. In the case of the neuronal plasma membrane, it's the complex of syntaxin and SNAP25 that we discovered here. In the case of any other acceptor membrane, an endosome, a Golgi, an endoplasmic reticulum to receive retrograde vesicles, and so on, the T snare would be a comparable structure, but composed of different proteins. But in the end, each would have its own snare. And so the snare hypothesis is very simple. The idea is you have VA and TA specifying vesicle A and its target for fusion. And VA binds TA, and you have VB and TB. VB binds TB. VA never binds to TB. VB never binds to TA. Therefore, you get A-type vesicles, B-type vesicles, and C-type vesicles. And if you add into this an A-type coat, a B-type coat, and a C-type coat that operate by the principles we discovered, you end up with selective cargo packaging and selective transport, which in a word is how it seems to work. I'll try to wrap up pretty quickly at this point. Just to bring you into the modern era, the proposal, we subsequently found, this is 1998, that the process was even simpler. You could do away with NSF, you could do away with SNAP, and you could take artificial synthetic lipid bilayers, putting the V snare initially vamped from the synaptic vesicle, and syntaxin in this membrane, and in another artificial synthetic membrane, the T snare, syntaxin SNAP25. And if you did that, they would automatically fuse. So that showed that the principle and the power of membrane fusion was encoded in the assembly of the snare proteins themselves. When VA binds TA, that not only targets the vesicle, it actually fuses them. What could be simpler than that? And along the same directions, to test this, there's now much evidence that establishes, indeed proves the essence of the snare hypothesis. But for me, the most satisfying as a biochemist, and probably the most compelling, comes from this experiment from the year 2000, in which what we did was essentially take the entire yeast genome worth of snares. So we took every single one of the snare proteins encoded in the yeast genome, and we expressed them as a separate protein. So we now have 20 test tubes filled each with one membrane protein. And now what we do is we mix these and put them into artificial liposomes, one containing a certain snare or set of snares, another a different one. For example, we put VA into one and TA into another. If we mix the two, we get fusion. If we put VA into one test tube, into one liposome, and VB and TB into another one, they will not fuse. In fact, we can test hundreds of combinations as if we knew nothing. Let's pretend we didn't know what was A, we didn't know what was B. We only had these genes. So we express the 20 genes, and we test them one at a time, two at a time, three at a time, four at a time, five at a time, in every possible combination between two lipid particles. And you know nothing in this, we assume nothing. And what we get is only a handful of combinations, and each of that actually work, none of the others work, and each one encodes or specifies the known transport route in the living yeast cell. What this proves is that the physical chemistry of these proteins encodes the specificity of vesicle trafficking in the living cell, at least in a yeast cell. So that is really very powerful evidence. So let me wrap up. I'm going to try to be very quick now. What do we now know about membrane fusion? As I've said, V and T snares will partner each other between membranes, and they form a bundle of 4 helices discovered by Jan and Brunger in 1998. As shown by them and others, including ourselves, this 4-helix bundle zippers up from one end to the other, and in so zippering, it actually pulls the 2 membranes together. It is that inner directed force that actually fuses the 2 membranes. Once the membranes have fused, the V and the T snare are now anchored in the same bilayer. And when they're anchored in the same bilayer, and indeed when their membrane anchors are assembled together to continue these helices, now fusion has taken place because a fusion pore has opened. What do NSF and SNAP and ATP do? Well, this is the lowest energy state. It's a fully folded 4-helix bundle. It is about 70 to 100 kT favorable energetically as compared to the unzippered or partially zippered snare complex. The result of that is that it takes a lot of energy to bust this up to allow the snares to be used again. NSF is a molecular chaperone, we found, that with SNAP protein as its adapter protein hydrolyzes ATP to using the energy to separate the snares to allow them to be reused. In the process, the snares are unfolded, and now they are in their high energy state. They then bind to each other specifically to form the snare complex, and in so doing, they not only produce their lowest energy state, but much of the energy that would otherwise be released as heat is now channeled to do work on the 2 lipid bilayers to transit them across their activation energy for fusion. So the power of pins, how does this work? It's actually very, very simple. Imagine 2 rubber balls, and we're going to stick 1 rubber ball at each end of a snare of a pin. Now, what I just described to you is actually a pin, a snare pin that forms between 2 membranes. And you'll see that this structure, with its inner directed force, looks very similar on a macro scale to what we see with 2 balls and a pin. Now, what happens if we allow this pin to insert into the 2 balls, each as a vesicle, and we stop the process at this stage? We have inner directed force, and what's going to happen to these 2 balls? Let's imagine the strength of this force exceeds the surface tension of these 2 balls. If it's 2 metal balls, nothing will happen. If it's 2 balls, each of which is malleable, like a drop of fluid, that's actually what a membrane vesicle is like. Well, what will happen is the force of the pin will exceed the surface tension, and in simple physics, the 2 balls will merge into one. At a physical level, it's so embarrassing to have spent 25 years, and you end up with this. I mean, can you imagine how utterly embarrassing that is? But it turns out not to be far from the truth. Here, actually, at a nano level, we have a single snare complex. We are pulling with tweezers via 2 glass balls, so-called optical tweezers. My collaborator, Yonglei Zhang, a young professor at Yale, is separating the snare complex forcibly, actually just doing the exact opposite. He's forcing the 2 ends that zipper last apart, and actually they will refold right in front of him. Zippered, unzippered, zippered, unzippered, zippered, unzippered. They fold, unfold, all or none. That is the power stroke of membrane fusion. And in a cartoon version of this, this is actually to scale. What happens is this is the V snare. The V snare is entirely unfolded before it interacts with the T snare. The T snare is not completely but largely folded into a 3-helix bundle, which then templates the membrane distal part of the V snare. There's the synaptic vesicle, or generally transport vesicle. There's the plasma membrane, or generally target membrane. The helix begins of the V snare zippering towards the membrane, and as it continues to zipper, the longer this membrane anchor, the connector between the helix and the V snare's transmembrane, gets shorter and shorter. If I have a rope, and I start turning it into a coil, I'm pulling in the rope. And in this way, the target membrane pulls in the vesicle. It does it typically with many snare proteins at a time, although in fact, though it's a little slow, one single snare pin is thermodynamically competent to fuse membranes. The vesicles are pulled closer and closer as the zippering proceeds, so close, in fact, that the water between the 2 bilayers has to be squeezed out in order for further zippering to occur, but further zippering does occur, and the water is then pressurized. We estimate, Frederick Poncet and I, that the pressure in here probably exceeds 100 to several hundred times that of the atmosphere, locally. At those sort of pressures, the lipid bilayer is no longer stable as the lowest energy solution to 4 lipid bilayers. Geometries with less water-exposed surface are required, including inverted micelles and soap-like structures such as these, which form. We don't know the exact structures, but under these pressures, surely the bilayer is dissolved, leading to a continuity or merger in an intermediate called hemifusion. But the process isn't over yet because the transmembrane domains haven't yet zippered. And as Fred Poncet showed together with my laboratory, the transmembrane domains continue to zipper, and these power the complete fusion and the opening of a fusion pore. And so, though there are many details that need to be understood at a physical chemical level, this seems to be the outline of how snare proteins deliver. So, to summarize then, what I've told you is that there's one general principle, the snare complex, that accounts for the specificity of an extraordinary range of delivery events that occur within a single cell and differently among many types of cells. The pathway of George Palade and its variants are responsible for the growth and division of the cell itself. Every time a cell doubles and divides, it needs to make an authentic copy of every compartment, A, B, C, and D. And the process that I've described to you makes that possible. Indeed, it is the process that generates the specificity of A, B, C, and D. And this specificity is encapsulated to a large degree in the physical chemistry of the snare proteins and the way in which they govern fusion and vesicle traffic. The secretory pathway, which is one aspect of this, takes on enormous significance for endocrine and exocrine physiology, generally for the secretion of hormones and growth factors, and imbalances in these pathways. Indeed, now we know many human disease genes that cause diseases related to metabolism, psychiatry, immunology, and the like. And finally, a yet further variant of exocrine secretion is the secretion of neurosecretion at every nerve ending, which I'll talk about in the third lecture of these series. What is remarkable to me today is that though these processes are extraordinarily different, they look entirely different. They are governed by different physiologies. They are governed by different signaling pathways. And indeed, though each was the province of a separate medical and physiological discipline, they all have underlying them a single beautiful general principle, which is such a wonder. And it really, I'd like to come back in closing to Buchner, is to me, I would never have imagined in 1980 when I started this work, 1978, that in so few years, it seems like a lot to the younger people, but to someone my age, it seems like a few years, as anyone my age can testify. It all goes very quickly, but I would never have believed in my lifetime that this level of breathtaking detail could be understood concerning a process that could only be observed in passing and crudely in living cells. And what one says about this process could be true of almost any process in modern cellular, molecular, developmental biology. It really is breathtaking, and I am in awe of it. What I am in awe of, increasingly, is not the scientist. I am in awe of the power of the scientific method. I am in awe of the discovery of the scientific method, because that is, the discovery of science is without a doubt the greatest discovery of science. And again, just in closing, back to Buchner, I found his words just really inspirational. We must never let ourselves fall into thinking that there are things we shall never know, because we should have every confidence that the day will dawn when even those processes of life, which are still a puzzle today, will cease to be inaccessible. And that was an amazing statement from this man who must have been absolutely an astonishing scientist. And finally, for critics, we all have them. I've had my share. I probably still do. They don't speak up as often. But I took this out of this quote comes from Johannes Kepler, and it's from the introduction to his great work, Astronomia Nova. It's translated from the year 1609, and these are great words. And he calls it advice for idiots. This is in his foreword, his words, not mine. Johannes Kepler, advice for idiots. Okay. Remember, Galileo had recently been out of prison, so it was that era. Okay. But whoever is too stupid to understand astronomical science, or too weak to believe Copernicus, he's thinking now about Galileo, without affecting his faith, I would advise him that having dismissed astronomical studies and having damned whatever philosophical studies he pleases, he mind his own business and betake himself to scratch in his own dirt patch. You know, as much as I would have liked to have known Buchner, I think I might have liked to have known Kepler better. So thank you. There are many dozens of people who've contributed to this project in my lab, probably about 200 over the years. I've mentioned the most principal people individually. And these are the folks that happen to be here to be with me on this day in October 2013 when the Nobel Prize was announced. You can see absolutely everybody was happy, even those who don't normally smile too much. And this is my wife, Joy, who is a professor of neuroscience at Yale and is here actually today. So I apologize to you for going over time, and especially to Alain, who has other things that he needs to run to. So thank you.